Today topic is structure of contrail proteins. Each actin filament is made up of two F actins, helically bound to each other like this. Troponin, tropomyosin and F actin. F actin. Two F actin helically bound to each other. Each F actin is polymer of monomeric G proteins. Each F actin is a polymer of monomeric G. G means globular protein. Globular actins. Two filament of another protein, tropomyosin. Tropomyosin also run in close to the F actin throughout its length, and a complex protein, troponin, is distributed at regular intervals on the tropomyosin. In the resting state, a subunit of troponin, masks the active binding sites for myosin on the actin filaments. Each myosin filament is also polymerized protein. Many monomeric proteins called meromyosins constitute one thick filament. Each meromyosin has two parts, a globular head with a short arm and a tail, and former being called the heavy monomeromyosin, and light meromyosin. Light meromyosin compose the hand and short arm project outwards the regular distance angle from each other of the surface, and is known as cross arm. The globular head is an active head, cross arm, actin binding sites and ATP binding sites. The globular head is an active and ATP enzymes head has binding sites for ATP and active sites for actin. Now the mechanism of the muscle contraction. Mechanism of muscle contraction. Mechanism of muscle contraction has been explained by the sliding filament theory. Sliding filament theory, which states that contraction of muscle fiber takes place by the sliding of the thin filaments, over the thick filaments. The muscle contraction is initiated by a signal sent by the CNS by a motor neuron. Motor neuron along with the muscle fibers connect to constitute a motor unit. The junction between the motor neuron and the sarcolemma. The junction between the motor neuron and sarcolemma of the muscle fiber is called the neuromuscular junction or motor end plate. A neural signal reaching this junction releases a neurotransmitter acetylcholine, which generates an action potential in the sarcolemma. This spread through the muscle fiber and cause the release of calcium ion into the sarcolemma. Increase in the calcium levels, binding of calcium with the subunit of troponin on the actin filament, and thereby remove the masking of active site of myosin. Bind with the actin site and remove the myosin. Utilizing the energy from the ATP hydrolysis, the myosin heads now binds to the expected active sites on active and form a cross bridge. Formation of a cross bridge. First, myosin filament ADP actin filament. Myosin head and cross bridge formation. The myosin head now bind to the exposed active site on the actin to form a cross bridge, and this pulls attached actin filaments toward the center of A band, and the Z line attached to the actins are also pulled inwards, thereby causing a shortening of the sarcolemma, that is contraction. During the shortening of the muscles is called the contraction. The I bands gets reduced A band retains the length. The myosin releasing the ATP, myosin releasing the ADP and P goes back to the relaxed state and ATP binds sliding rotation ADP and P ATP myosin filament. Myosin had the cycle of the Kris. The ATP is again hydrolyzed by the myosin head and cycle of the cross bridge formation and breakage in repeated causing further sliding. The process continued till the calcium ions are pumped back to the sarcolemma cristina, resulting in making of actin filaments, and the Z lines back their original position, that is relaxation. The relaxation. On the other hand, some of the muscles possess very less quantity of myoglobin and appears pale or whitish. These are the white fibers. Number of mitochondria are few in them, but the amount of sarcoplasmic reticulum is high. Depends upon anaerobic process for the energy. It means the mechanism of the muscle contraction divided into three parts. One is excitation, second mainly four parts, four step: excitation, contraction, coupling, and contraction and relaxation. And energy source for contraction, energy source for the contraction. ATP provides the energy required for muscle contraction. ADP released during contraction ultimately change into the ATP by creatine phosphate. In the muscle, formation of ATP take place by the oxidation of the glucose and fatty acids. In red muscles, myoglobins can store oxygen which take part in the formation of ATP. In anaerobic condition, lactic acid get deposited in the muscles and muscle feel painful conditions. This is the whole mechanism of the contraction of muscles. In next video we are start in the next part of skeletal muscle. Thanks. Have a nice day.